I unfortunately woke up with a 100-degree fever this morning, so I won't be able to join you in person and have to be here online. I can see that Kevin attracted audience from all over the world based on the participants that dialed into your defense. So I think today we can expect to see a very energetic and informative presentation. Kevin's thesis work relates to how bacteria express their genes in response to the different environments they live in and the different stresses they are adapting to. He also has written a software package, which many people find enviable in its quality and presentation. So a package called iModulon DB that you'll probably see during the presentation that is getting use around the world. He will also talk about his work on Bacillus subtilis, which is a key industrial organism, and how its transcriptome can be understood. And he's also probably going to detail very complicated adaptation responses of Escherichia coli to temperature and to ROS stress, so reactive oxygen species stress. I would say personally, it's been an absolute pleasure to work with Kevin as a graduate student, always enthusiastic, energetic, and informative, as I said earlier. He's a highly talented young scientist that's going to give us a great defense today. He has mastered writing very complicated documents, presenting difficult and intricate results with great clarity. So let me say also that this is a public defense at the University of California. We have it open to the public, and once Kevin has completed his presentation, it's open to question from anyone present. But after that, we will move into a closed session with him and the committee. So take it away, Kevin.
Awesome. Thanks so much, Dr. Palsson. So here we go. I'll share the screen. Okay. Everybody hear me all right? So thanks everybody so much for coming. I'm Kevin Rychel. This is my PhD defense. And today we're going to unravel the sensory systems of cells and begin to understand the regulation of gene expression. So I thought I'd start the story at the very beginning. I was born in 1996, and at the same time, so was genomics. So this is one of the first ever genomes, and you can see that thin black line going all the way across. That represents the DNA of a bacterium, and then each of those colored arrows represents a gene. So when this happened, when this came out for the first time, you could really understand a cell from this genome scale, whole perspective, enumerate each and every one of the genes that make it up and all the pieces of this system. Dr. Palsson has a really great story about this. When he opened up the journal Science and saw this for the first time, he realized that this whole cell understanding is actually possible. So he dropped all the research he was doing before. He flew from the University of Michigan over here to UCSD, and he started chasing this systems biology understanding. And by 3 years later, by the time I was 3, he had done it. So this is a genome scale metabolic model that him and his student created. So they took each of the chemical reactions encoded by that genome, and they laid them out into these metabolic networks, which you can see along the side. And then they were able to use a mathematical tool called flux balance analysis to calculate what each of those reactions is doing, given the growth objective of the cell and the environment around it. And so what was really interesting here is even though you have the complex biology, all those reactions, the system takes on a smaller number of states. So you can map it down into these little regions on this graph here and actually understand the state, understand the system more easily in a smaller number of variables. So this was really inspiring to me as a way to bring this genome scale understanding into something that a human being can actually understand. Maybe not a 3-year-old, but somebody can. And so we're building towards this genome scale understanding, again, taking the genetic basis of life, DNA, and converting that into these metabolic models, which can trace food sources, chemistry, growth, energy, byproducts, all throughout the cell. However, these models are incomplete. there's something, the cell doesn't use the entire metabolic network all the time. Each of these reactions is subject to changes in gene expression, which can up or down regulate different reactions, use different parts of this network in different times. And the thing in the middle that is sort of mediating all of this is called the transcriptional regulatory network, or TRN. So what we're looking at here in these ovals are regulators, which are things that can sense the cell's environment and the cell state and respond to that. And then each of these circles represents a gene, which is regulated by those regulators. And genes that are regulated as a group are called a regulon. And typically these groups of genes have the same function or similar functions and are needed under the same conditions. So there's been a lot of effort to try to understand the TRN and how it really works. But for my whole life, it's kind of evaded us as a field as far as how to model this effectively. We did get some progress, again, when I was still 3. So here we have one of the first transcriptomic data sets. And this, in this image, each pixel represents a gene. And then the colors represent whether it's up or down regulated in green or red. And so we, again, we're looking at transcriptomics, but again, at the genome scale. And this really tells us what the cell is doing under whatever condition you're measuring it in. So you could expose it to different antibiotics, different stressors, whatever condition you're interested in, you can now see each and every gene expression change. And at the time, this was heralded as a new kind of map, but it's difficult to read. Again, there's a ton of variables. It's really complex. So we realized that this was a pretty important type of technology. And by 2007 or 8, we had developed RNA sequencing, which is a different way of generating transcriptomics that ties the cost of transcriptomics to DNA sequencing, making it a lot cheaper. And it's also more accurate than microarrays. And then by the time I hit my Bieber years, the National Institute of Health realized that it's probably a good idea to store all of these in the same place and make them reusable and accessible to people. So they created the Sequence Read Archive, or SRA, which stores all the raw data in one place. And then by the time I entered college, we had a few thousand bacterial profiles available. And then by the time I graduated college, we had explosive growth, a tsunami of public data. So there's all this RNA sequencing data. And this really creates a new opportunity for me as I'm entering grad school to try and look at the transcriptome from this new kind of large data perspective. So traditionally, the way we would look at the TRN is to focus on a single system. We want to pick out an individual regulator of interest, upregulate it, knock it out, see where it binds on the genome, look at all these phenotypic experiments. This is super important because this establishes a gold standard baseline for how each of the individual pieces of the TRN. However, when you try to make models of the TRN globally based off of these annotations, they've often not been super predictive of data. It's been really difficult to work with these annotations in general. So there's an opportunity here. Now that we have all of these data sets, we can use that existing data to infer regulatory interactions at the genome scale. And this requires minimal new lab work. It's data-driven, of course. And so this is really what I wanted to chase in my PhD. So the idea is to take all those transcriptomic data sets and infer the TRN. What we really want here is to know the structure, so which genes are co-regulated with one another, and also to be able to interpret these data sets. So there's thousands of genes, and we would rather look at a smaller number of knowledge-enriched variables. And then we also want to be data-centric, scalable, reusable. We want to make sure that as that tsunami continues to wash over us, we're still able to use the same method. And so the lab's answer to this question is independent component analysis. And here, what we're looking at is a gene expression data set X, which is each of the genes by each of the samples that we took. So we expose a bacterium to a lot of different conditions. And then we break that up with this machine learning method called independent component analysis, which is an unsupervised machine learning method originally developed for radar and sound signal processing. But the way it works here is to actually break it up and separate the structure of the TRN. So which genes are co-regulated in that M matrix there. And then separate that from the activity, which is what each set is doing in each sample. So we call each of these co-regulated sets of genes that are inferred with this method, we call them iModulons, or independently modulated sets of genes. And the general workflow here is to take in-house data or public data that we downloaded from that SRA database. And we align them to the genomes, do our normalization and pre-processing. We create a gene expression matrix, and then we break that up with ICA. And with the N matrix here, we can threshold that to get our co-regulated gene sets and compare those sets against the known regulons in order to understand which groups are new and which groups are previously known, previously explored. And the differences between our iModulons and the known regulons give us opportunities for discovery. And then on the A matrix side, we can do statistics actually on the X matrix or the A matrix, but when we do it with X, we get lots and lots of gene changes. When we do it on the A matrix, we get a smaller number of more easily interpretable variables. So the prior work here was from my mentor, Dr. Anand Sastry, who applied this method to the PRECISE 278 data set that stands for Precision RNA-seq Expression Compendium for Independent Signal Extraction with 278 samples. So here he found that actually a lot of the iModulons that were pulled out matched very well with the known regulators. In some cases, they matched even better than the prior work. And he was also able to track the activity and see quantify changes from transcription factor knockouts, mutations, etc. So this was a really great motivating study that validated that iModulons do work, and maybe we should try them on more and more organisms. So the strengths here, we can use data alone to globally summarize gene expression. And this matches really well with known regulation, better than any similar method, according to a study by another lab. And it uses matrix decompositions. So we get this structure where we can calculate the explained variance. We can infer the activity for new samples easily. And it's just generally a great thing to work with. It also handles batch effects very well. So we can normalize those away, or we can pull out technical iModulons that reveal those batch effects. It also does have a few limitations. So you have to actually activate each of the regulators that are in the cell. And we need around at least 50 conditions in most cases. You want to release in duplicate. So you do have to get a fairly large data set in order for this to work. We also have a couple of assumptions that aren't necessarily true. It assumes independence of the underlying regulators. So in real life, the TRN regulates itself. There's a lot of interactions between regulators, and those might create some unusual artifacts in the iModulons. We have really complex regulation. It doesn't always capture it perfectly. It's also linear, and sometimes the actual TRN is not linear. But in general, when we have nonlinear interactions, we get multiple iModulons, which sort of capture that curve shape. And I might show you a few of those later. And then it is somewhat sensitive to a few parameters. So the thresholding and the dimensionality selection, we wrote extra algorithms to optimize those. But in general, the iModulons are very robust. So these parameters don't change the overall story. So the three things that sort of were left to be done that I've worked on in this PhD is one, to show that iModulon analysis works in another organism. And instead of just validating and making sure that it matches well with what we expect, I want to answer new biological questions and really make this accessible and interesting to a microbiologist. And then two, I want to develop an online knowledge base of iModulons. So we realize that this tool is widely applicable. It can tell us anything about any changes to gene expression. And so it's a really valuable thing to put in the fingertips of others. And that's what aim 2 is about. And then in aim 3, we're going to use this framework to understand TRN evolution and integrate it with other technologies. So instead of just characterizing them and focusing on the iModulons, we want to actually use them and apply them as a tool. And so those 3 aims turn into 5 sections for this talk. So I'm actually going to go through it in this way, where in aim 1, we're going to characterize specifically Bacillus subtilis, which is a really interesting model organism. And then in aim 2, in sections 2 and 3, we're going to develop and expand iModulon DB, which is that online knowledge base. And then in steps 4 and 5, we're going to do 2 different evolution studies. The first one to paraquat, which creates oxidative stress, and the second one to high temperature stress. And so I'll start going through those. The first one is this Bacillus subtilis TRN. And so Bacillus is this scientific model for sporulation. You can see sporulation there, where this stress-resistant spore is forming inside a bacterium. And then it's also a model for biofilm formation, those things you see in your shower that you have to scrape up. Competency, which is taking DNA up from the environment. And then it's an industrial organism that's used for protein production. When you buy protein products, sometimes they're made with this organism. It's also a microbial food. So if you ever had this Japanese fermented bean dish called natto, it's made from this. It's also the first genetically engineered probiotic was made from Bacillus. So it's a really informative and interesting organism. And the TRN is fairly well characterized. There's an online database full of the known information about that regulatory network, which means that it's a great place for me to start and compare what do the iModulons say versus what it's known. There's also this great Nicholas 2012 data set. So there was a microarray with already 104 diverse conditions, which is again a great starting place for ICA. So that data set that I just mentioned is X over here. Again, it's all the genes by all those 104 different conditions. And then we used the ICA pipeline I presented to pull out the iModulons, which again are those independently modulated groups of genes that you can trace their activity across each condition. And so then what I basically did in this project was go through each of those iModulons and try my best to understand what is it doing, why was it pulled out as a regulatory signal, and does it make sense with what's known? Does it provide any new insights? And what I was able to do was assign functions and categories to each of them. And you can see the categories here in this donut plot in the top right. There were 11 that had to do with carbon sources, so different foods the bacteria can eat, or amino acid synthesis, building up the proteins, stressors, biofilms, toxin production even, sporulation. So as someone who had never studied bacteria before, this was a really great way for me to get an idea of basically everything that the cell can do. And it was a really fun and interesting project to work on. So here's an example of an iModulon. This is kind of a simpler one to get you oriented. This was the FADR, or fatty acid utilization iModulon. So on the left, you can see there's a set of genes, all shown in that tan circle, which match really well with the known FADR regulon. And they're all genes which are needed to consume fat. And so what was also really interesting is if you trace the activity of this iModulon across all the samples, you find that Bacillus subtilis is a lot like us. When we have glucose in our blood, or in the media in this case, we will use glucose as our energy source. But when there's no glucose around, when you starve it at that hour zero mark, all of a sudden you activate fat utilization. You try to burn fat. And so, and then once you use up all the fat that's available to you, then you downregulate this. So we can really easily and simply trace what this entire group of genes is doing, rather than go through each gene individually. We see it all at one activity level. So a lot of the iModulons worked a lot like the fatty acid iModulon, where they made sense, everything seemed to match with the literature. But in these 8 cases, and in a few others, we saw unexpected things, relationships that maybe weren't obvious or were suggested by a few papers in the literature, but not directly studied out. And with the iModulons, you answer questions you didn't know you were asking when you generated these data sets. So we could pull these out as hypotheses and suggest to the field to go explore them further. And so you'll see relationships between stressors like ethanol, metabolites like tryptophan. All of these different lifestyle choices are changing things. But here I'm going to talk about this one. So we found the LEX-A iModulon, which contains all of these DNA repair genes. And it's activated whenever DNA is broken. It senses broken DNA. So we saw in the hydrogen peroxide and mitomycin conditions an activation of this iModulon, which makes sense because those are conditions that damage DNA. So we expected an activation here, but unexpectedly, we also saw swarming cells activated this iModulon. So swarming is shown in this movie in the bottom left. There's this central colony, and then swarming away from it, you have all of these Bacillus cells trying to find a new home, basically. And there's nothing inherently DNA damaging about swarming. You wouldn't really expect this to cause DNA damage. So I looked into both swarming and this iModulon to try to figure out why might this be. And so I proposed a possible mechanism. The first part comes from this paper, which showed that certain cells within that central colony are going to accumulate oxidative stress naturally. And those cells which accumulate that stress are going to, 1, activate the DNA damage response, and 2, stop producing biofilm. And then if we look at another paper in the literature, we can see that biofilm and swarming are in a trade-off. So piecing these two stories together, we find that the cells which activate Lex-A are also going to be more likely to swarm. And this provides a nice, interesting hypothesis and really reveals why that thing in the data set might have occurred. And so I went through all of those iModulons. I went piece by piece, went through them. And then I also was interested in going another way, which is looking at conditions and globally across all the iModulons, how are those changing? So Bacillus is a model for sporulation, which you can see spores here. Those are those smaller round circles. Those are highly stress resistant. You can dry them out, you can boil them, and once they're in a more favorable environment, they'll still germinate back into Bacillus. And you can see some cells forming spores there. And the cells will form spores whenever they're particularly stressed out. So in this case, there's this amino acid starvation condition over 8 hours, which causes sporulation. And I was really interested in how globally does the entire transcriptome change across this process. So in the first hour, you can see this activation. I don't know if my laser pointer is very visible, but you can see the first hour activates all of these amino acid synthesis genes, which are just trying to make up for that starvation. And then in the second hour, when that doesn't work, when we're still starving, we enter this community response phase where we transition to stationary phase. We start producing toxins and bulking up our biofilm, trying to protect ourselves. We take up zinc, which is a major cofactor for sporulation. We even do things like cannibalize each other. It's a really interesting step where the bacteria are kind of just freaking out. And then in the third phase, we activate each of the sporulation iModulons one by one. And so it's a really nice overview and summary of this entire process that, again, is in a smaller interpretable number of variables. And was also interesting, there were some new things. These two metabolic iModulons were not known to be the food sources for the late stages of sporulation, but they popped out as signals in the data. So it might be that these extracellular energy storage molecules are actually being used to power the last few steps of sporulation. So the conclusion for this study, so we did demonstrate that iModulons do indeed work on a new organism on the other side of the bacterial phylogenetic tree in this gram-positive organism. And this motivates analysis of more organisms, and it also provided those 8 or more insights about diverse processes, including the DNA swarming one I mentioned, and also 2 that were later validated in metabolic models that I didn't discuss. And then we can globally summarize this major process of sporulation. And I think that these iModulons could be really informative for any research into Bacillus, including applications in probiotics, biomanufacturing, or even with sporulating pathogens. So given the success of both the original E. coli study, this iModulon, this Bacillus study, and then a third Staph aureus study, we decided to make iModulon DB, which is this knowledge base of microbial transcriptional regulation. And the idea here is to further FAIR principles. So in data stewardship, you really want to make sure that your data and knowledge is findable, accessible, interoperable, and reusable. And in the past, in the first few studies, we had these PDFs. So you can see here that fatty acid iModulon I showed you with some graphs about its genes, its activity across every condition, some notes about its regulation. But this isn't great because there's limited page space. You can't interact with this or update it. There's no, the data access requires you to be able to code, and sharing is really cumbersome. So we thought we could do better. And so we actually developed this online knowledge base to make iModulons as fair as they can be. And so I built it. This is iModulondb.org. So I'll go ahead and show you. So here's a list of all the organisms which we've analyzed with iModulons. And I'll go ahead and jump to Bacillus, which is the one I just spoke about. And so here on this page, if it loads, there it is. So these are all of the iModulons, each and every one of the signals that was found in that transcriptome. And you can read functions for each one, some statistics on them. And so if we go ahead, and of course, there's a link to my paper. If we go to one of them, so this is the SIG-D iModulon, which regulates motility. So that swarming that I showed you. And it has all of these flagellar subunits. So all the flagella that are needed for swarming are listed, all the genes for that are listed here as members of this iModulon. And you can also see, interestingly enough, so there's this column here with SIG-D. And so this means, the green check means that it is known to be regulated by SIG-D. And in a few cases, you actually find unknown genes which were not known to be regulated by this regulator. But the iModulon analysis suggests that they are co-regulated with motility. And so these would be interesting for someone who wants to characterize new genes and find new functions that might be related to this. And then we can also look at the activity profile going across all of the samples, and we can interact with all these graphs, of course. And what we can see, for instance, we can also color by the media or whatever metadata we have. And so here we can see that motility gets turned off during sporulation, which makes sense because why would we sporulate? Why would we be going around when we're busy sporulating? And then we can also see some comparisons between the iModulon and the known regulator, and so forth. There's also this great search functionality. So I can show you, for instance, searching for a metabolite of interest. You can see which iModulons are associated with it, and you can follow each of the genes associated with that as well, and see which iModulons those genes are in. In this case, this gene wasn't in any iModulons, but you could go and learn more about it there. We can also download everything at this download link. And I wanted to highlight too this about page, which one brings you through what iModulons are, and also links to our code bases. So you can see here, this one is used to generate iModulons from scratch. And actually, if you go to this PyModulon code base, we can analyze iModulons in Python. And specifically, I wanted to point out this step 3, inferring iModulon activities for new data. So if you have generated an RNA-seq for any of the organisms we have iModulons for, you can go and compute your own iModulon activities here, and then go back to iModulon DB and look up those important iModulons and learn more about them from that source. So what we've done is taken that original matrix decomposition, and we pulled the M matrix structure, and we made that gene row showing all of the genes. And then the A matrix activities, we pulled those out and showed you that activity bar plot going across the center. And then if it's annotated with a regulator, if it seems to match these known biological signals, you can also find information about that. And so this really puts iModulons at your fingertips as any researcher. And so for each of these sections, I have I'm going to present a first author paper, except for this one here. So these are all papers that I contributed to and worked with. You can see the first authors listed here. And so this was really the expansion of iModulon DB and how we built it up from just the 3 original organisms to a much larger resource. So the way we did this was to actually go back to that tsunami of data, that big opportunity I mentioned for this current time in systems biology. And we went to the SRA database. So at the end of 2022, there were 60,000 bacterial profiles. And we went through all of those. And we found that quite a few of them weren't annotated well enough with strain information to really know exactly which organism they were analyzing. So we couldn't really use those. And then for quite a few organisms, we also didn't, they were rare organisms that didn't have enough data for us to analyze with the iModulon. But for these last 10,000 samples, we actually pulled them out and analyzed almost all of them and published on most of them as well. So I assisted here by generating the iModulon DB dashboards, helping to mentor quite a few of these projects, edited the drafts and so forth. And what we end up with is an iModulon DB that really spans the entire web of life. So here in the center, you can see the last universal bacterial common ancestor. And branching out from it is the evolution of all of the rest of the diversity of bacteria that we see in the world. And each of these blue circles represents an organism that's been analyzed on iModulon DB. So even if you're not a scientist, you might recognize tuberculosis, salmonella, staph. There's cell factories like Pseudomonas putida. There's even an extremophilic archaea, which really loves to live in acidic, really high temperature environments. And so iModulons are really informative about such a diverse range of life. And I also want to highlight Precise 1000. So this was, again, not work that I first authored, but this is a large E. coli data set. I mentioned in the introduction, we only had 300 samples. At this point, when we were expanding iModulon DB, we ended up with 1,000 E. coli samples. And this data set is available on iModulon DB, really gets us closer and closer to that quantitatively irreducible representation of the transcriptional regulatory network. And this includes samples that are going to be analyzed in the next few studies. So I thought I'd bring it up now. So iModulon DB is constantly growing. We have 11 organisms on there now, and we have 5,000 high quality samples, 1,700 iModulons. And what I'm really proud of is this corner down here, where I show that iModulon DB is continuing to be used throughout the community. So we gain about 7 new users every month, and they tend to stick around. So last month we had 367 unique users, and this is becoming a tool that's used by the community. And so again, iModulon DB enables the search, visualization, and download of all these iModulons. It contains diverse organisms and insights, like all those that showed you for Bacillus. It has a, it relies on FAIR principles because we download all that data from the internet, and we kind of return the favor by providing these quality controlled, contextualized, and knowledge-enriched data back, so that people can really have a lot of new information at their fingertips. And this paves the way for studies that use iModulons. So rather than focusing on just characterizing them, now in this last section, I'm going to use them in combination with other tools to learn new biology. And so for this first section, we're going to talk about the evolution of paraquat tolerance. But actually in both sections here, so the rest of this talk really, we're going to talk about how iModulons interoperate with other methods. So we use laboratory evolution, genomics, physiology, metabolic models. And so I'm going to present the data, and I want to make it clear that I am inferring connections and coming up with putative mechanisms which relate across all of these. And I find them to be coherent and informative, and I think they're correct, but this is important to mention. These are inferences in a lot of cases. And so fundamentally, we want to know, how does the TRN evolve? And specifically, we're looking at 2 different stressors. So I mentioned in this section, we're going to talk about paraquat, which is an oxidative stressor. And in the next section, we'll talk about high temperature. And these have some interesting applications. So we want to design cell factories that produce products for us. And oftentimes those cell factories are going to experience stresses like oxidative stress, high temperature, and so forth. So we want to make sure that we can design stress tolerance into these strains. And we could use iModulon changes or mutations from these studies in order to improve those designs. So with the evolution of paraquat tolerance here, we did laboratory evolution, specifically a tolerization adaptive laboratory evolution. So we start with a small amount of stress, and we ramp it up over a long period of time, which generates these strains that are highly stress tolerant. And then I take that strain, or these strains, and I do a multi-level genome scale systems analysis on them. So we take all the DNA mutations, all the iModulon changes, and then we even use metabolic models and phenotypic measurements, and kind of relate between all of these to understand how were those strains able to tolerate so much stress. And then what we end up with are these tolerance mechanisms, which are stories within all of that data that explain, in my opinion, how the cells were able to do that. So the stress that we're talking about here is oxidative stress, or ROS, or reactive oxygen species, ROS. So these are chemicals which pull the electrons off of cellular components. And it's the same thing that causes rust. You're probably familiar with hydrogen peroxide, which is a pure form of ROS. And this is relevant to normal metabolism. So ROS are formed on accident by cells all the time. And it's also a major chemical weapon used in immunity. So if you have an infection, you're going to dump oxidative stress into that infection. And it's a frequent problem again in bioproduction. So we want to engineer better cell factories that don't have to deal with this quite so much. And another great thing about it is we have the oxidized me model, which is a metabolic and expression model, like those ones that I introduced at the very beginning with Dr. Palsson made around us 3. And so this is a state-of-the-art model from 2019 that incorporates all of the major stress mechanisms of ROS. So ROS will damage iron, and they will target different enzymes, damage DNA, and the sensitivity to it is different in different reactions. So that will lead to changes in the entire metabolic network. And we don't usually work with ROS in the lab because cells tend to behave very strangely when you give them hydrogen peroxide. So instead, we use a common herbicide, paraquat. And how this works is it will find its way into the cell through a transporter. and then it will donate an electron to oxygen, and that will create these evil glowing red ROS agents. And those will then go on and cause all of the stress and damage to the cell. But then paraquat will actually strip an electron from the cell's normal electron carriers and steal this electron to then reinitiate that cycle and continue pumping out more and more stress inside the cell. And what I've shown here are each of these letters, which represent the different systems that have actually mutated and changed as a result of the evolution. So they touch on all of the elements of the paraquat cycle. And what is tolerization and adaptive laboratory evolution? Well, we start with this low amount of stress, as I mentioned. And in this case, we're using a pre-evolved version of E. coli, which can just barely grow inside that low paraquat base. And once it fills up the flask, we have a robot here on the 4th floor of this building, which will pass that into a new flask. And if it grew fast enough, it will actually up the amount of stress that the cells are getting. And so by the end of this, you can see going across, we go from a low level of paraquat with cyan color to a pretty high level of paraquat with purple. And what we also did was pull out a few of these flasks and do DNA sequencing. So we were able to see which genes mutated. And interestingly enough, in all of the 3 first-generation ales, we saw the same 2 first genes mutate, EMRE and ACE, which suggests that these are really important genes for tolerating the stress. And then we continually accumulate more and more mutations, ultimately getting to that stress tolerant phenotype. And what does the stress tolerance look like? Well, you can see in black the starting strain, which with just a small amount of paraquat stops growing almost immediately. So this is a really intense stress for that. And then in the evolved case, the blue line, you can see much more paraquat tolerance. It can grow at much higher levels. Interestingly enough, though, there is a trade-off. The cells are making a compromise because the condition at 0 stress has a lower growth rate. So we're not able to grow as well without the stress. So I can go through the mutations and look at all of them and how they seem to have affected the genome. And what I've done is basically assign each mutation to its closest gene. And you can see the boxes in that image are sized by how many times that gene mutated. And then we get categories out of this. So the transport category was probably affecting how paraquat is able to get into and out of the cell. And then we have mutations in central and energy metabolism, which that's upstream of the electrons going into the paraquat cycle and might actually stop this entire cycle from happening, or at least reduce the amount that it's occurring. And then we also saw a bunch of iron-related mutations, which could affect how the stress is actually affecting the cell, or actually use iron sensitivity to ROS to regulate those phenotypes. We can also look at the iModulons. But before that, I actually used this more typical transcriptomic comparison, which is differentially expressed gene analysis, going through each and every gene and seeing which one is up or down regulated in the comparing the starting strain under stress and the evolved strain also under stress. And when you do that differentially expressed gene analysis, you get 1,774 changes. So this is too many for me to go through and analyze. This is that problem that I mentioned in the introduction of not being able to interpret all of these things. But after we run ICA, so we use this as part of that precise 1,000 data set, and after we do the iModulon analysis, we see only 42 differential iModulon activities, or DMOs. So this is much more tractable for me to actually go through each one and try my best to characterize globally what are the transcriptional changes that occur. And so there's 2 main categories of iModulons I wanted to highlight: the stress and growth changes, as well as the metabolic changes. So I drew this knowledge graph, which connects the DNA mutations to the RNA iModulon changes to metabolism and phenotypes. So here we're making inferences, trying our best to connect between the changes to each of these different levels of biology. And what we result in, knowing that the stress has changed and this evolution has occurred, we get to these different phenotypes, which reveal possible strategies that actually can overcome the stress. Okay. In the first case, we have this sort of simpler change. There was this gene EMRE, which I mentioned was the first gene to mutate. And in 2 of the 3 strains, it got copied 42 different times in the genome. So you get a whole bunch more of the gene itself, which in the transcriptome leads to an upregulation of an associated iModulon. contains the same genes as the genes that were copied. And then why was this chosen? Well, my best guess, why was it selected for? My best guess is that EMRE is able to pump out paraquat. And there's this was known in the literature, this was worked on before in the 90s. So we knew that, so we now can see a pretty clear relationship between all three of these levels, pumping out paraquat being the ultimate goal and phenotype for this mutation. And so in metabolism here, we are rewiring metabolism to decrease the paraquat cycle. And so I mentioned that second mutation was ACE. This is in pyruvate dehydrogenase, which is that entry into the TCA cycle. And we also saw quite a few other mutations in the TCA cycle, which is a little initially surprising to me because the TCA cycle is the major energy producing pathway within cells, and mutations tend to be damaging. So why would it be that the cell under stress is actually shutting down the system or decreasing its flux? And the answer comes down here. So I think that NADH is usually used to produce energy, but in the case of paraquat, it will actually be used to produce more stress. And so this will lead us to, if we do decrease the TCA cycle, I predict that would decrease paraquat cycling and lead to less paraquats instead of metabolism by using this pathway a bit less. And the iModulons actually show a shift towards anaerobic genes, which are also not using as much NADH. So this is reinforcing that same mechanism and leading probably to a decreased use of the TCA cycle. We also in the iModulons see an upregulation of glycolysis, which is going to be an alternative energy producing pathway that will make up for this. And we can, since this metabolic work was so interesting, we actually used that oxidized me model that I had mentioned to study this. And we can strain the model using all of the RNA-seq levels that were relevant, as well as the phenotypic measurements that I'm going to discuss in the next column. And when we do that and strain these models, we see that indeed the optimal flux for this case is a decrease to the TCA cycle. And the NADH that is produced by the TCA cycle has a less steep slope, so it's less responsive to small changes in paraquat, and the cells cannot grow at higher paraquat cycle fluxes, which is probably going to help us to decrease the amount of stress we're actually getting. And then phenotypically, we can make this arrow type plot, which here what we're looking at on the y-axis is the biomass yield, so how efficient are these cells being? And you can see the starting strain with no stress is very high on the y-axis because it's very efficient. But then we have the glucose uptake rate here on the x-axis, which shows how much fuel is the cell really using. And once we add stress to the cell, you'll see that it drops to this bottom left corner because it's very ineffective because of all the damage, and it can't take up as much glucose because it's just so busy dealing with all that stress. But then after doing the laboratory evolution, we see a shift only to the right. So we're upregulating the glucose uptake, but we're not becoming more efficient. And that's because we've damaged the TCA cycle. We can't become more efficient, but we are able to grow faster just by using more and more glucose. And then finally, so after the success of those two mechanisms, the efflux and the metabolism and a few of the other ones, we end up with a cell that is much less stressed out after the evolution. And we also see a lot of iModulon activities, which basically seem to just be sensing how much that stress is really has changed. And so I was able to infer this middle level of phenotypes, which are probably they're the things that the iModulons sense, and they're likely to change as a result of this decrease in stress. So we have iron sulfur cluster damage. So leucine and biotin are two iModulons are two metabolites, and the synthesis pathways for those are really sensitive to oxidative stress because they contain these iron sulfur clusters, which break under stress. And after evolution, we see that those are downregulated because the metabolites themselves have increased their levels, and that leads to basically going back to normal for this. And we also see changes to general growth and stress, these master regulators, as well as a few other things like DNA synthesis, cysteine, and so forth. So in this section, we showed that a global analysis of iModulons can reveal these interesting tolerance mechanisms. I talked about upregulation of efflux, rewiring of metabolism. iModulons can infer sensory inputs from the TRN, so we can see those changes to the iron sulfur clusters we can see all of those effects on the transcriptome and quantify them more easily now that we have this new mathematical framework. And then mutations from this work were actually employed by a collaborator over in Denmark to develop a oxidative stress tolerant strain. And these mutations did seem to have a benefit for growing in oxidative stress on their own, as opposed to just in the evolution. So in this final section here, we're going to talk about the evolution of high temperature tolerance. And here we evolved E. coli to grow at 45 degrees Celsius or 113 Fahrenheit, which is a lethal temperature for wild type cells. And this was a follow-up to another study. You can see these purple strains here, which were all grown at 42 degrees Celsius. And one of those samples was actually then further evolved, in my case, to get these 6 high temperature tolerant strains. And in high temperature stress, we get protein misfolding. So high temperature is just atoms moving around faster and faster. And when that happens to proteins, they'll often unfurl, and the sticky centers will then stick to one another, and you get these aggregates. That's how cooking works. That's how your meat gets stiffer. And it also involves a lot of changes to enzymatic rates. So different parts of metabolism are going to respond to this. And the biology here is relevant to things like the fever response. So you up your own temperature when you have E. coli infecting you. And global warming, of course, is going to raise the water temperatures around the world. So it would be really interesting to know how bacteria are going to evolve to tolerate that. So I wanted to go through all of the genetic changes that occurred in these strains. And I showed you in the oxidative stress how you could do that and categorize each of them. Here, it became really difficult because across the only 6 strains at 14 last time and way fewer mutations, here we have 500 total mutations. There's no particular category of mutations that seems to be overemphasized or enriched. So what happened here was this mut-L gene mutated. That is a DNA repair gene. And so we end up with hypermutators, which just can't repair their DNA as well. So we get this huge diverse changes to the genome, which is useful for E. coli in a lot of ways because it allows them to explore that mutation space and find new strategies. Not useful for me because I don't know which mutations are important, which ones are just along for the ride. So instead, I can look at the iModulon activities. So here we show, again, the wild type at high temperature versus the evolved strains at high temperature. And we can see which iModulons are changing. We also see that only 20 iModulons capture about 60% of the variants. So we can really see only a few clear changes that actually had a huge effect on this phenotype. So I again drew a knowledge graph, and so you can see on the far left, you might have to squint here, I know things are a little small, but high temperature is on the far left there, and that leads to a few other inputs like the general stress response or thermosensing regulators, and then that's inputs on the far left that's covered up, and then we have a column of mutations. So these are the mutations that appeared to be relevant based on the iModulon analysis. So these are regulatory mutations that would be upstream of these iModulons. And then we have the iModulons in the center, changing and they're sized here by the explained variance. And then on the far right, you see downstream of those iModulons, what are the effects, the possible pathways and strategies that these cells are using to be able to grow at high temperatures. And I'll just briefly say what these 3 main strategies were. We streamline the stress response. So we turn down general stress and we upregulate that specific heat stress tolerance mechanisms via by RPOH. And then we have a large protein export. So it seems that protein export systems are being upregulated, which this is worth studying further. I don't know for sure, but I do think that it's possible that these misfolded protein aggregates are coming out of other transporters within the cell. And then we can streamline metabolism as well. So there are different, we actually, again, move more towards fermentation, which might avert auto-oxidation or do other things that make the metabolism more efficient in this high temperature case. So instead of going through the details of these mechanisms, I thought I'd show these activity phase plans, which reveal basically here what we've done on each of them is plot one iModulon against the other. And we also include these kind of low opacity, sorry, in the room here, it might be hard to see, but these low opacity points are the other samples in precise. So the black lines represent the general trend that we usually see across all 1,000 data sets. But then the hop tail samples, the evolved strains I'm talking about, those have their own trends, which are actually a bit separate from the traditional trends. And that might be because of those mutations in those regulators. So we're actually shifting, for instance, on the far left, we had a mutation in RPOS, that general stress response regulator, and that pushed us to a downregulation of RPOS and caused the trend line to move so that we basically avoid that general stress response and grow faster. And then we also had changes to motility, redox and aerobicity, iron uptake. And so you can see both the temperature trend and the possible effect of these mutations. So in this section, we showed that iModulon analysis reveals these clear evolved mechanisms and hypermutators. And I mentioned those three key patterns. So streamlining the stress tolerance, exporting large proteins, and then streamlining metabolism. And finally, we have these activity phase plans, which leverage those hundreds of other samples in order to reveal the usual trend and then the evolved new trend that changes to regulation that happen. So in conclusion here, we characterized the Bacillus TRN. I showed you those more than 8 novel relationships between stressors, lifestyles, and metabolites. We globally summarized sporulation. We were able to tell this transcriptomic story with a smaller number of variables and reveal new things about sporulation as well. And then we had dissemination via the development and expansion of iModulon DB. So this is an online resource that has been widely adopted in the community by over 300 people each month. And we analyzed over 11 diverse organisms. And we also then evolved. So in the last section, we evolved strains to tolerate paraquat and high temperatures. And I showed you how we can use changes to genomics, phenotypic measurements, metabolic models, and ultimately get this coherent picture at this new kind of systems level genome scale for how all of these changes are occurring. So we were able to successfully take transcriptomic data and infer the transcriptional regulatory network. And some of the themes that kind of popped out that I highlighted in the conclusion of my dissertation, iModulons are a middle layer of regulation. So a lot of times when people analyze transcriptomics, they only think about the downstream effects. Changing this gene is going to have what effect? But upstream, there's all these sensory and regulatory inputs that control gene expression. And it's really important to also consider those upstream systems and really understand what cells are sensing and changing. And then we had this genome scale dataset-wide perspective, which revealed all of these new global insights and interesting hypotheses. But we didn't have enough time to go into the wet lab and test every single connection. And so it's important to do follow-up studies and things like that metabolic modeling study in Bacillus or the oxidative stress engineering study. These are really important. I highly encourage those, and I love to see their results. So it's definitely part of this quest for new understanding and not the entirety of it. And then we have FAIR principles. Again, we reuse that online data and we enabled the fairness by providing back these curated knowledge-enriched TRNs. So this work shows the value of separating the M matrix, the structure, from the A matrix, the activity of the transcriptome. And iModulons are an excellent way to compute this from data alone. We got these transcriptome scale insights and made them accessible to everyone. In the future, I do think that there's an opportunity. Well, there's quite a lot of opportunities here. We can do iModulon comparisons. I didn't mention it on the slide, but we can do iModulon comparisons across different organisms, and that can reveal how evolution has happened kind of in the natural world at this large scale at the TRN instead of the genome. And then we can also think about reformulating M to maybe address some of those limitations and assumptions which aren't quite true in ICA. So we have a lot of value to the iModulons as they are, but we could incorporate network architectures, nonlinear methods, maybe even kinetic promoter models. And we want to make sure that we recapitulate the insights that we observed in the iModulons, but we also can maybe find ways of incorporating other data into these as well. Cool. So I wanted to spend just a moment on the dedication and acknowledgements here. So I wanted to mention all 4 of these people. Jeff Donaldson was my best friend from high school. And he really taught me how to ask big questions and be curious. So my grandma taught me that where there's a will, there's a way. And I definitely leaned on that a lot during this process. Stephen Calderon was a good friend of mine who was really just always in awe of science and tried to keep that for myself during this process. And then Nate Penn was my father-in-law, and he just raised the man who really helped me through everything and was just a real joy to everyone from that. And I also want to acknowledge everybody. So Dr. Palsson was super inspiring from the moment I first met him. And I'm so happy to be part of his legacy and to have worked with him and been in this lab. Dr. Anand Sastry was the person who enabled everything that was in this work. He developed iModulons and recruited me here at the coffee shop right outside. So a big thank you to him. Dan, Adam, and Patrick, all of you were really important mentors, especially Dan throughout this process, just being there, providing all the insights that you did and really helped me to get through this. The ales that I worked on, so Justin and Ka did the ROS and the high temperature ale before I took them over and added the iModulon piece to them. So a big thank you to those guys, as well as the wet lab researchers. So Ying and Richard, dry lab research can't happen without wet lab research. So a big thank you to you. I want to thank my funding, Novo Nordisk and the Center for Biosustainability. All my lab mates, we had a really great culture here at the SBRG. I really enjoyed working with all of you and the committee. Thank you to you guys. I collaborated with each of your students at different times, and we had a lot of great fun working on those projects. My other collaborators, especially those watching from Europe, I really appreciate it. It's been really fun and exciting to be able to work internationally across borders during these last 5 years. And then I want to thank my family and friends, Sean for literally giving me the shirt on his back, and my parents and everybody else. Sorry, I'm getting a little sappy here, but and everybody who I grieved with, everybody who's been there for me, distracted me, kept me focused, all those things. Really appreciate all of you. So with that, I do have a QR code here. If you want to scan it, you can actually get links to all of the papers that I have written, details about them, and contact me that way as well. But with that, I want to thank everybody for listening, and I'll take any questions.
Thank you for that, Kevin. That was a truly energetic and informative presentation. So well done. We should move into the Q&A session. So there are a lot of people online here from all over the world that can either type out questions into the chat box, or if they want to ask them in person, they can do it too. But I'm at a disadvantage. I can't see who's in the room and who's raising their hands and wanting to ask questions. So you want to run the questions yourself, Kevin, or should we ask a member of the committee to do that?
I can do it. That's fine. I'm up here.
Hey, Kat, early on in the presentation, I think it was, is it okay now? Okay. People couldn't see me on the Zoom.
Oh,
We didn't want to interrupt you, but it sounds like it's okay now.
oh, worked out. Oh, I'm sorry about that. Oh, yeah, you could interrupt me.
Good.
Nice.
Hey, Kev, I'd like to know how all this, chat about, AI and everything, how that affects where you see kind of, research like this happening moving forward.
Yeah. Well, ChatGPT is just going to change the world in so many ways, and these large language models are really going to be a big deal. I've been really interested in them personally, and if you ask ChatGPT, they know some things about iModulons, but they also hallucinate a ton of them. You ask it about iModulon DB, it knows all about it, can tell you all the features. You ask who made it, it says somebody else, which of course broke my heart. But yeah, I think that there's a lot of opportunity there to form, filling in these predictions and inferences that I've been making. I think that AIs, if we can get them to a place where we trust them and they can cite their sources more effectively, it could abstract a lot of this away, for sure. But yeah, you're still going to need people with this expertise to check on it all and all of that. The models themselves are also potentially really useful for specific machine learning things. So I mentioned maybe reformulating that structure of the TRN around promoters. There's an opportunity for the same types of models that underlie these large language models to understand the TRN through their own, through that algorithm instead of an component analysis. So yeah, definitely a lot of work to be done there. Thanks, Adam, yeah.
Yeah, so a lot of the RNA-seq we looked at are kind of community, large number of cells for transcriptomic analysis. There's variability across the different strains, right, in the population. And we thought much about, I guess, more sequencing methods towards, single cell analysis or looking at the stochasticity across, a growing population just within a flask. Thoughts on that?
Yeah, I mean, I definitely there were a few cases even that swarming case where you see within the colony itself that there's some cells that are getting the oxidative stress and some cells that aren't. I think it'd be really cool. I always wanted to actually do a single cell RNA-seq project as part of this PhD. Maybe I'll do it after this at some point in my life. But yeah, I think that a lot of the assumptions that we make about regulators being about inferring the regulator activity, really we're inferring the average regulator activity across trying to basically that stochasticity is built in there. And so yeah, it'd be much more accurate to go single cell, really looking at what each thing is doing. And in the ROS study, I spent a long time on that project, actually trying to go through the differences between the strains. And I found that it was too noisy. It was really hard to do. The main insights, like the ones that I showed today, occurred in all of the strains, and that was the easiest thing to do. But you do get pretty wide error bars in those iModulon activities, and it would be better to get a more detailed look. But honestly, most of it was not very clear, not very interpretable. So I think still you want to get to those kind of aggregate larger changes are still going to be the easier ones to find. But maybe if you start with higher resolution data and then find those further down the line instead of scooping up a bunch of cells and analyzing them all together, that would be, yeah.
It's interesting to think about.
I agree. Yeah, Dan? Thanks.
I think we've gone farthest maybe the complex posture to ultimately studies differently to
Yeah. Well, in the hot tail especially, there were a few samples that were we just want to make sure we characterize all of our controls across diverse conditions, and we have those intermediate steps. I didn't spend much time analyzing the intermediate steps, mostly because it wasn't fully covered. There wasn't everything. I could see one midpoint and not the rest of the midpoint. So I was like, well, I'm not going to spend time on that. It would be cool. It would be great to get each mutation's individual effect. That was a dream in a study that I designed, and then we realized it was just too expensive to use CRISPR to make all these mutations after the fact. But yeah, if we did a little bit of RNA-seq after each mutation, you could directly attribute the effects versus the mixture. Like in the hot tail, there were the hypermutation thing really made it difficult to draw those connections. I could only do it when I was pretty clear, oh, this is a regulatory gene, so it probably is causing this. Yeah, I think having more density of that. And you don't need necessarily actually going across every temperature or every paraquat concentration. You really need just the stress case, and you want to make sure that stress case is reflective. I think that the stress used in the paraquat study was maybe a little lower than it should have been, because the stress is less, we're less stressed out signals were stronger than they probably should have been, because they actually used a slightly lower stress level. It can be difficult to, these strains are really finicky. It's really difficult to actually go back and reanalyze them and re-put them in that same condition. It's better if during the ale we collect more and more stuff. Of course, it's just hard to know during the ale exactly when you need to be doing that. So it's a challenge. And I think what I did here was able to get around a lot of those challenges, but it would be definitely better to do that. yeah, freezing a bunch of cells as you go across the L, I guess, is the way to do it.
I think Anna has his hand raised.
Ah,
Hey,
hey,
I had to make it through Zoom, but great presentation. It was really exciting to see everything that you've done and all you've accomplished in these past years. Yeah, one question that I had is, you've built up iModulon DB to have all of these expression profiles, and you showed that cool plot of like how it's been increasing over time. It's going to keep increases, it's going to keep exploding. Like, how do you have a plan on how you're going to keep it updated?
Yeah, actually, funny enough, there's 2 new species. Thank you to Kira and Brahm, if you're watching, for waiting, because I had to do all of this. I couldn't update it with 2 more species. So we're actually it's continuing to grow right now. And every time we do a pull from the sequence read archive, we find more organisms that have just crossed over that threshold to now be analyzable. I think that, well, for one, the code for updating iModulon DB is going to be packaged really nicely. I've actually inherited certain databases where it was difficult to update them. I'm going to make sure that whoever is updating iModulon DB after me does not have that problem, and it's very simple. The dream is to just drag your iModulon JSON file right into a hard drive, and then boom, iModulon DB is updated. I have most of the code to make that happen, but we just got to set it up. So yeah, I think that there's definitely, we definitely have more interest in this, and it's also a great master student or even course project. This past year for Bioengineering 212, Dr. Palsson's course, we assigned people to just choose an organism of interest and see if you can get the iModulons for it. So yeah, I think we can do more work in working with like other labs outside of our own to use there was somebody analyzed a pathogenic strain of E. coli, and they did the iModulon analysis, but they didn't necessarily use our coding framework. So it would be good to do some more outreach on that front as well. But yeah, we'll continue working on the concrete plan. I'm sticking around for a few more weeks at least, so we will figure out exactly what the future of it is.
Yeah, that's really exciting.
Yeah, thanks. Well, and thanks for having the idea. We still have on the whiteboard at the lab the original wireframes for it. I was like, you can erase everything but those. Yeah, maybe after I leave they'll go away, but I'm not sure. Any other questions?
Actually, I have one more if
Sure.
we have time. Yeah, so with the oxidative stress experiment, so EMRE was a paraquat, it was specific to paraquat, and it seems like maybe the NADPH also had something to do with paraquat itself. If you used a different type of stressor, do you think you'd still see a lot of the same mutations, or do you think, or a different type of oxidative stress? Do you think you'd see the similar mutations, or do you think that some of them would be different?
I don't think we would see similar mutations. I think, so I showed you that, here, I can jump back to that, load this. So here in these mutations, so the transporters, so the EMRE is not necessarily fully specific to paraquat, but it wouldn't be, you wouldn't see that for hydrogen peroxide, for instance. And the central and energy metabolism, I think, really mutated because the electron carriers are the target of this paraquat and they get stolen, So I think that the left side here, these transport energy metabolism things, might be a bit specific, especially the transporters. For the energy, NADH is often associated with oxidation. So we might see a few of these energy metabolism changes. But I do think, and actually in that oxidative stress tolerance study where they used variety of oxidative stress stuff, what I suggested they use is these iron mutations, because these ones are the downstream predicted effects where it is actually after the oxidative stress. So yeah, I think a lot of the mutations are actually trying to just prevent the oxidative stress from even forming this A and F over here. And yeah, that was one of I originally thought it was a design flaw because this was handed to me as just a pure ROS study and my mechanisms were very specific to paraquat. But at the end of the day, I think you would see that with any kind of herbicide or any, any kind of antibiotic that you're using. And by mapping these back to their mechanisms, we can sort of predict which ones would be informative for other oxidative stressors versus this one. Yeah.
Yeah, that's really cool. Get rid of the stress before it even happens.
Exactly. Yeah. I mean, yeah, evolution's pretty smart somehow.
Thanks.
Yeah, you're welcome. So we do lose this room in 15 minutes, so it might be best to enter the closed session. If we do need more time, we can go, the committee and I can go upstairs. But for everybody else, thank you all again so much for being here.
Yeah, thanks for attending. So we'll move into the closed session then.